Good morning and good evening, everyone. So welcome to this time Immune seminar. So today is my really pleasure to introduce our speaker, Wenwen Zeng from Tsinghua University Institute of Immunology. Wenwen actually got her bachelor degree also from Tsinghua, and then she further pursued her PhD in Texas, in University of Texas Southwestern, with James Chen, study innate immunity. And then after she finished her PhD, she took sort of a interesting detour. First of all, she joined Genentech as a postdoc for 2 years, which afterwards she joined New York Rockefeller University in Jeffrey Friedman's lab. I guess that time she's studying the immune neuron interaction, which actually she's developed her own program after she got independent. Wenwen got independent from 2014 in Tsinghua University, and then she started her own program. As I mentioned, she mainly focused on immune neuron crosstalk, regulating metabolomic program in vivo. And then I think it's a quite fascinating field, which also grow very, very rapidly. When we got different awards, just to give a few example, 2015, she got National Young Investigator Award, and 16, she got Outstanding Young Scholar Award, 求是 Science and Technology, funded from 求是 and Science and Technology Foundation. 2018, she got Young Investigator Award, and 2018 also, she got the National Science Fund for Excellent Young Scholar. I think for the past couple years, Wenwen actually review and publish quite a few significant work, as I mentioned, related to the immune neuron crosstalk, how they control and regulate the metabolic program in vivo. And I guess it's also today's her topic. So without further ado, Wenwen, please take over. We are looking forward to your talk.
Thank you. Thanks, Tran, for the really kind invitation. And also this introduction is extremely nice. And also I want to thank Lee for organizing the seminar series as well. So our lab has been studying the peripheral nervous system, especially how the peripheral nervous system control the peripheral organs and how the peripheral neurons respond to the peripheral insults. So this is what we have been interested in the lab. So on one side, we're interested in how the brain control the peripheral organs. Particularly, we are studying the sympathetic nervous system, and we have been using the adipose tissue as the model organ to study how the central nervous system activity control the peripheral metabolism. And on the other hand, we're also studying how the peripheral nerves respond to the peripheral insults. Particularly, we have been studying how the central nervous system respond to the peripheral injury. We use the injury model to study the neuroimmune response. So the reason we use the adipose tissue as the model organ is due to its really plastic nature. For instance, in mice, you would see the really heterogeneous distribution of the adipose tissue across the whole body. For example, in the normal trough diet-fed mice, you will see the adipose tissue underneath the skin, inside the peritoneal cavity, as well as many as well as around many organs. Some of them are white and some of them are brown colored. The brown colored ones have more mitochondria, are highly vascularized, and more energy expenditure prone. Well, in response to chronic environmental cold, you would see browning of the white adipose tissue. Now you will see many of the white adipose tissue, they actually turn to brown. So they are highly thermogenic in this case. While in the obese condition, for example, high-fat diet-fed mice or genetically obese mice, you would see the whitening of this adipose tissue. And this heterogeneity has been observed in the human population as well. You would see some of the human population, they have more metabolic active adipose tissue, for instance, this case. And in the obese subjects, you would see less energy expenditure adipose tissue. They tend to store a lot of triglycerol in the adipose tissue. So the energy state of the adipose tissue can affect the systemic metabolism quite profoundly. For example, the major cell type in the adipose tissue called the adipocyte, in the fasted state, the adipocyte would release the free fatty acid to provide energy, while in the fat state, the adipocyte would process energy and store the triglycerol as in the form of lipid droplet. While the adipocyte can also function as a really critical endocrine organ. For example, leptin, adiponectin are among the adipokines, which are released by adipocytes and influence the systemic energy state. And also the adipocyte can also release lipids, metabolites, and many molecules to the organ in the systemic level. So the adipocyte can affect the systemic metabolism. So as I mentioned in the early slides, the white adipose tissue can undergo many changes in response to environmental energy states. For instance, in response to cold, you would see beiging of the white adipose tissue. So this beige or beige fat or browning bright fat, they actually can undergo lipid hydrolysis, undergo mitochondrial biogenesis. So they have a lot of mitochondria, so they highly thermogenic, so they can consume energy quite efficiently. While in the obese condition, you would see whitening of the adipose tissue. So the adipocytes are larger, so they contain a lot more the triglycerides. And within the tissue, the immune cell can undergo dysfunction. So you will see the inflammatory cytokine upregulation and the adipokine change as well. So leptin, adiponectin level, they will change. So they will affect the systemic energy balance. So in this process and in the highly plastic change of the white adipose tissue, we are particularly interested in how the neuronal activity may regulate the white adipose tissues. For instance, whether the neuronal activity can regulate the cold-induced beiging, or under obese conditions, how the neuronal activity may adapt. Or the question, so we had this question for a long time. So one reason we didn't know how many neuron fibers were actually present in the white adipose tissue was due to the challenges in imaging the neural innervation in the adipose tissue. For example, in the brown adipose tissue, which actually has the highest distributed nerve fibers, you would see only very few fibers using the classical frozen or paraffin sections and the immunostaining. You will only see very few fibers surrounding the blood vessel. For example, the arrow and arrowheads on the tissue section. So this really leads to question about how many nerves are actually present in the white adipose tissue. So many people actually believe there are not much. So we started this journey by adopting the whole mount staining procedure at that time point. So in this case, by using the whole mount imaging, we can actually see the axon bundles of only a very few axon bundles within the white adipose tissue. This is in the length of 2 centimeters back a few years ago. So we start to see the nerve bundles, and they're especially in their intact form, not just the fragmented axons. So the question still remains. So we get further to study what kind of anatomic structure actually present in the white adipose tissue. Then we further delineate what are the functions of the nerve distribution in the white adipose tissue plasticity. So we adopted the whole mount staining procedure later. So this case, we established the protocol for staining the white adipose tissue based on the iDisco procedure. This way, we can actually using synaptophysin to visualize the nerve fibers across the whole inguinal fat pad. So in this case, this is in the length of 2 centimeters. So synaptophysin actually labels synaptic vesicles that said you actually have very dense synaptic vesicles within the nerve fibers within the tissue. So we also use the other markers, for example, PDP 9.5, a pan-neuronal marker to stain the nerve fibers. So this is the inguinal lymph node within the fat pad. So you will see that in the whole inguinal fat pad, you will see really dense distributed nerve fibers. So basically, they are all the axons projecting into the tissue. So we started to characterize these nerve fibers by using different markers. In fact, we found the majority of them actually stand for tyrosine hydroxylase positive. So basically, they are sympathetic nerves, and they are quite densely populated within the whole tissue pad. So we also use the co-staining of tyrosine hydroxylase together with the adipocyte marker. In this case, we use paralipin. So the blue color labeled paralipin within the tissue. So when we quantify the pattern of the nerve fibers and the paralipin labeled adipocytes, we actually can see more than 90% of the adipocytes can be directly regulated by the nerve fibers from the 3D perspective. So basically, the previous study claimed there might be 10 or 20% of the adipocytes which can directly regulated by nerve fibers. And our data actually indicate pretty much almost every adipocytes can be directly controlled by the nerve fibers. So basically, the signal from the central nervous system to the peripheral nervous system can be quite precise in this way. We can also use EM to label to identify the neuron fibers on the tissue. So in this case, we can see the synaptic vesicles in this nerve fibers. And this is a lipid droplet filled white adipocyte. So in this way, in response to the sympathetic neuroactivity, we can imagine that the neurotransmitter from the synaptic vesicle can be released that can signal to the white adipocyte to initiate the signaling events. Then we start to ask what are the functions of the nerve fibers in different biological processes. For instance, the cold-induced beiging of the white adipose tissue. So this fat pad is after 7 days of cold exposure. In the co-staining of tyrosine hydroxylase together with the UCP1, we actually can see the UCP1 positive beta cells are quite spatially colocalized with the tyrosine hydroxylase labeled nerve fibers. So that led us to hypothesize for two. So one is the neuron activity might be able to directly regulate the beiging process, and the second is once the beta cells occur within the tissue, their activity can be directly regulated by the sympathetic neuronal engagement. So firstly, we localize the neuron cell bodies to further understand those nerve fibers. So in this case, we injected the retrograde labeling marker CTB into the fat pad. So we can detect the CTB labeled neuron cell bodies across the cilia ganglia and also the synaptic ganglia chain. And those neurons actually can be labeled by track A. So basically, during development, they use track A signal. They respond to the ligand of track A called NGF for their survival and axon growth. So we delete track A by crossing TH-CRE to drive the deletion of track A in the sympathetic neurons. We can largely genetically deplete the innervation of sympathetic neurons within the fat pad. So this allows us to further study the function of this neural innervation in the adipose tissue biology. So in this case, we use the cold-induced beiging model. We can see that in the control animal from room temperature to cold challenge, the white adipose tissue actually undergo beiging process. You will see the occurring of the multilocular beige cells, while in the when the sympathetic neurons are genetically ablated, you will see this beiging process is compromised. And so on the mRNA level, you would see the Beijing-associated genes, for example, UCP1, DIL2, CIDA, PDC1 alpha, their expression is all compromised. So further, we use the pharmacological depletion to further confirm the local innervation of the sympathetic neurons is indeed critical for code-induced Beijing. So we use 6-hydroxydopamine. We locally apply 6-hydroxydopamine to a fat pad. So we can really get rid of the locally distributed neurons in this case. Then we challenge these mice with cold exposure. So in the control treated animal from room temperature to cold challenge, you would see occurrence of these multilocular beige cells, while when the nerve fibers are pharmacologically depleted, you would see that the Beijing process is really compromised. Well, this process can also, the defect of the Beijing can also be confirmed on the protein level of UCP1. So when the nerve fibers are depleted with 6-hydroxydopamine with time going on, the upregulation of UCP1 is really compromised. While also on the mRNA level, the UCP1 as well as other genes, their upregulation is also compromised as well. Well, on the other hand, we also confirm the local innervation is really a dominant source of the neuronal activity in regulating Beijing. So in this case, we remove the adrenal gland by surgery procedure, the adrenalectomy. Then we challenge the mice with cold exposure. So the sham-treated animal, they can undergo the Beijing normally, while in the adrenal gland removed animal, you can still see Beijing. This is also confirmed by protein level of UCP1 as well as iron level of UCP1 as well as others. So basically, the adrenal gland is not the dominant source to drive the Beijing process in this case. So on the receptor side, so the sympathetic neurons basically use the catecholamines to signal to the adipose tissue to drive the Beijing process. Well, on the adipose side, they highly express beta-adrenergic receptor to respond to the neurotransmitter. So in this case, we use beta-adrenergic receptor to phenocol mice, and then characterize their Beijing process. So in the control animal, we would see that the Beijing can occur in the wide adipose tissue showing the beige cells, while in the beta-adrenergic receptor to phenocol mice, the Beijing is largely compromised. And also on the protein level and as well as MR level, we would all see reduced Beijing, showing reduced protein UCP1 and reduced Beijing associated genes. So basically, the sympathetic neurons signal to the beta-adrenergic receptor, then drive the Beijing process in response to the cold exposure. So we think the role of sympathetic neurons is conserved between mice and human, because when we characterize the human sympathetic neurons, we can detect they are predominantly distributed by the PDP 9.5 and tyrosine hydroxylabeled sympathetic neurons. So basically, in the human fat pad, you would also expect the regulation of the wide adipose side by sympathetic activity. When we image the sympathetic neurons in the adipose tissue, we found that they are not static. So basically, they are highly dynamic. In the pathological conditions, for example, in the high-fat diet induced obese animals or OBOB, the genetically obese animals, you can see that the density of the sympathetic neurons decreased quite dramatically when we image the obese and diabetic mice, either in the diet-induced obese or genetically obese. So basically, that means in under pathological conditions, the central nervous system really lost control of the local control of the wide adipose tissue. Well, on the other hand, under physiological conditions, for example, in response to chronic cold exposure, we actually can see the upregulation of the neural density. So for example, comparing the room temperature and the cold challenge, we can see the neural density actually change a few folds with the time going on. So basically, the neural fibers increase the density to cope with the environmental challenge for the thermogenesis. And this process is reversible. So when the animals were put in cold for 7 days and then put back to the the thermoneutral condition, 32 degrees. So with time going on, you actually can see the neural density decrease again from 1 week, 2 weeks, and with time going on. So we can quantify the neural density. For example, the cold exposure, you would see increase of the density, while the thermal neutrality condition, you would see decrease of the density. And this neural density change is highly correlated with the Beijing-associated genes, including UCP1 as well as others. So basically, the fat pad really adapts itself to deal with the cold temperature challenge. And when we look into the cellular and molecular mechanism of the neural density change, we found in response to the chronic cold exposure, the axon fibers actually undergo the regeneration process. So in comparing the room temperature and cold challenged animal, you can see the increased STM2 signal within the fat pad. And by quantification, we can also see the STM2 signal increased quite significantly. While on the cell body, we can also see the STM2 signal is increased, meaning those neurons really undergo axon regenerating process. So they basically they regenerate more axons to innervate the local fat pad. And so we looked into the neurotrophic factors which can regulate this process. So basically, the ligand for TrkA, NGF, can be induced in response to cold exposure. So basically, that can signal to the TrkA signal to lead to the axon regeneration. So the NGF is critical for the sympathetic neuron development. So when we culture the sympathetic neurons in vitro, and then we add NGF neutralization antibody, we can see the axon growth is inhibited. While post-developmentally, because NGF knockout is lethal, so we use the NGF neutralization antibody to transiently block its activity. So in response to cold temperature change, we can see the room temperature and the cold challenge can increase the density. While when we use the NGF neutralization antibody in a transient manner, we can see the neural density change is actually can be greatly reduced. So and also on the receptor level, so TrkA, the phosphorylation of TrkA and the retrograde transportation of phospho-TrkA from the axon to the cell body is indicator of its activation. So in this case, in room temperature versus the cold challenge, we can see the cold challenged animal, the phospho-TrkA signal in the cell body is increased. So basically, the signal from NGF to TrkA is engaged in response to cold exposure. And further, we use the TrkA mutant form generated early by Dr. Gente's group. So this mice, because the TrkA receptor is engineered so it can susceptible to an inhibitor, NAPP1. So in this case, when we challenge the animal, we can see the STM2 signal in the neural cell bodies. When we apply the NAPP1 to pharmacologically inhibit TrkA signal, we can see the increase is compromised. And this is also reflected by the neural density as well. So basically, when we inhibit TrkA signal, we inhibit the axon regeneration and the neural density change is compromised. And this has a consequence on the Beijing process as well. So comparing the room temperature and cold, we have this Beijing. And when NGF neutralization antibody is applied to the animal, we have reduced neural density. We also has impaired Beijing process on the HE staining as well as determined by qPCR as well. And also when the TrkA receptor is inhibited, we also see reduced Beijing on the HE staining as well as qPCR level as well. So by far, I have showed how the sympathetic neurons regulate the Beijing process. While in the wide adipose tissue, in addition to the sympathetic nerves, they're also highly vascularized. So basically, there's really dense blood vessels in the tissue, as well as many other immune cells as well to maintain the tissue homeostasis. So we then further looked into the vasculatures in the adipose tissue. We can verify that really in the inguinal wide adipose tissue per cubic millimeter, there's about 1 meter of vasculature. While in the broad adipose tissue per cubic millimeter, we have 14 meters of vasculature. So this tissue is highly vascularized. That actually can enable the quick exchange of energies and molecules between the tissue and other organs to sustain the energy balance. Well, of particular interest to us, we imaged the tyrosine hydroxyl labeled sympathetic neurons and the CG31 labeled vasculatures. We can actually see that more than half of the vasculatures actually are co-aligned with the tyrosine hydroxyl labeled sympathetic neurons. So basically, they align with each other. So the spatial distance actually indicates they can have reciprocal crosstalk between these two systems. So one process we study is angiogenesis process, because the angiogenesis has been observed by a few groups. So in response to cold challenge, so this is isolectin labeled vasculatures. So in response to room temperature, we can see that cold challenge actually can induce angiogenesis process. Is more vascularized in response to cold. Well, in this case, when we locally ablate tyrosine hydroxyl labeled synaptic sympathetic neuron, we can actually see the angiogenesis process is compromised. We can determine the reduced angiogenesis process by quantifying the density of vasculatures. So on the molecular level, we found actually the sympathetic neurons can promote the production of VEGF alpha. So basically, the sympathetic neural activity induce the level of VEGF alpha that actually promote angiogenesis process. And also under obese conditions, this process is actually is impaired. The alignment of the sympathetic neurons and the vasculatures is defective. So as I mentioned earlier, in the obese condition, for example, genetically obese, in the OBOB, DBDB, or high-fat diet-induced obese, we would see that decreased tyrosine labeled sympathetic neuron. Well, in this case, we also have less vascularized adipose tissue. And also, when we quantify the vasculatures with sympathetic fibers, we can also see that in the obese conditions, the vasculatures with close distance with the sympathetic neurons, they actually are greatly reduced as well. So that means the regulation between the nerves and the vasculatures are lost in the obese condition in this case. So in the adipose tissue, there are many cell types. For example, we can also see the CD45 labeled immune cells across the whole tissue. So right now we are working on how the sympathetic neurons, they can crosstalk with many other cell types. For example, the CD45 positive immune cells, and even within the immune cells, there are really heterogeneous population of immune subtypes. So we try to understand more of the intracellular crosstalk between the sympathetic neurons and those other cell types as well. So in this part, I have showed you the neuroregulation of the wide adipose tissue biology. So firstly, we revealed the dense sympathetic innervation in the tissue, and then we delineated some of the functions of the sympathetic neurons. And then lastly, we found that there's really physiological and pathological changes of the neural innervation in the tissue. So the second part, I'll show you how we study how the peripheral neuron, they may respond to the peripheral insults by studying the neuroimmune response. So the sensory nervous system gives us a nice model organ to study the peripheral neural response to the nerve damage. For instance, in response to nerve damage, there's a lot of studies showing the neuroimmune response actually contribute to the neuropathic pain. So in this case, so under normal condition, when you have increased stimulation, you would have increased pain intensity. However, post nerve damage due to the contribution from neuroimmune response, you would have the allodynia and hypogesia. So in this case, you have a lower threshold to detect pain or highly sensitive to the stimulation. So we use the sensory nervous system, basically to study the which regulate the limb, and with cell bodies on the DRG, the dorsal root ganglia, to study the neuroimmune response. So in this case, we introduce the sciatic nerve injury to the peripheral axons. We actually can see the infiltration of the immune cells into the dorsal root ganglia. So they are very close to the cell bodies in this case. So we can label the CD9B positive cells. So this is before injury, this is after injury. Really, the immune cells accumulate within the nervous system. So we started to characterize the neuroimmune response. So really, we didn't see other cell types. For example, CD3, B220, CD11C. So this is quite specific to the CD9B immune cell infiltration. So we try to understand more of the immune response by using the RNA profiling. So post injury, we actually can see some of the chemokines, including CCL2, 7, 12, and cytokines IL-6. They are upregulated in response to the nerve injury in the dorsal root ganglia. So basically in the where the cell bodies are located. So those chemokines can be responsible for recruitment of the CD9B positive cells. So when we look into which are the cell types expressing these chemokines and cytokines, we actually can see highly expressed chemokines and cytokines in the neural cell bodies. So basically those neurons, they actually initiate immune response. They upregulate those chemokines and cytokines in response to the nerve injury. So we start to characterize the pathways which mediate the upregulation of those chemokines. So this way we use an in vitro model. We culture the neurons in vitro. We introduce axon injury in vitro. In this case, we can actually detect upregulation of those chemokines CCL2, CCL7, and CCL12. While some other cytokines, for example, IL-6 is not changed. So I'll mention in my last part, IL-6 actually rely on other cell types for its upregulation. But CCL2, 7, and CCL12, as well as CSL1, they actually can be upregulated by axonal injury by an intrinsic mechanism of the neurons. So we use the SHRI screening to identify the pathway which regulates this neuroimmune response. So in this way, we use the selected pool, for example, including many of the molecules responsible for the innate immune response. For example, MyD88, NEMO, RIG-I, MD5, those are all the signaling molecules involved in the innate immune response in response to, for example, pathogen infection. Well, when we do the screening, we actually found one hint coming out, which is SOM1. So when SOM1 is knocked down, the CCL2 or CCL12, its upregulation is compromised. So SOM1 actually belongs to the same family with MyD88 and TRIF. It is third domain containing protein family member. And we all know that MyD88 or TRIF, they are involved in many of the signaling pathways mediating cytokine or chemokine upregulation in response to pathogen invasion. Well, SOM1 is actually quite specific to neuron expression. So that in this case is actually responsible for the neuroimmune response, so we can use in situ hybridization to confirm expression of SOM1 in the neural cell bodies, and also using Western to show that SOM1 is quite highly expressed in the neural cell bodies, and it's gone in the SOM1 knockout mice. So when we do the shRNA knockdown of SOM1, just in a few cells in the DRG neurons, for example, the AAV also encode TD tomato reporter. So in this way, we use TD tomato to label the shRNA knockdown cells. In this way, we can actually see the genes mediated by SOM1, so CCL2, CCL7 is actually decreased, meaning there's an intrinsic pathway in the neural cell bodies, in the neurons, which mediate the CCL2, CCL upregulation by SOM1. So on the SOM1 knockout mice, we can confirm the role of SOM1 in mediating those chemokines. So when we introduce the nerve injury into the SOM1 knockout mice, we can see those chemokines, CCL2, 7, 12, they're all reduced. Well, CSF1 seems rely on redundant pathway for its upregulation. Well, in the SOM1 knockout mice, we can also see the CD9B immune cell infiltration is greatly compromised. Or the family member of SOM1, TRIF or MyD88, we found those are not required for this process. For example, in the MADAT knockout or TRIF mutant mice, we can see those chemokines and cytokines, they can upregulate normally. And the CD-LAMB immune cell infiltration are also normal. So basically, the role is quite specific to someone. And we delineated the signaling pathway that someone actually signal to DLK to jump to phosphosyndrome, which has been shown in the axon degeneration pathway, so someone can regulate this process. So in this way, we can see someone actually signal to DLK when we use someone knockdown. The DLK phosphorylation downstream of the pathway is defective. So in response to neural injury, we can quickly detect the DLK phosphorylation within a few minutes. However, when someone is knocked out, the DLK phosphorylation is greatly reduced. When we cross the DLK with Wnt1A to delete the DLK in the sensory neurons, we can also see the chemokines and cytokines are greatly reduced. Well, there's redundant kinase for DLK, so you would see reduced level of those chemokines or cytokines. And also infiltration of the LAMB cells is also compromised as well. So in this way, the DLK actually has some effect on CSF1, meaning actually DLK function downstream another molecule, which is redundant with someone in this pathway. Well, downstream DLK, we can see that JNKs are activated. When we use JNK2 and the JNK3 double knockout mice, we can see the chemokines and the cytokines are reduced, including CCL2 as well as others, and the infiltration of CD-LAMB cells is compromised in this case. And also on the transcriptional level, JNK actually active phosphosyndrome. And in the someone heterozygous animal, we can see the phosphorylation of syndrome when with the time going on post the injury. While in the someone knockout mice, we can see the phosphosyndrome is greatly reduced. And also when we delete syndrome in the neurons, we can see the induction of those chemokines and the CSF1 cytokines are reduced. Well, by using ChIP-seq of targeting syndrome, we can see that syndrome actually directly recruited to the promoter or enhancer region of the cytokines and chemokines, including CCL2, 7, and 12, and CSF1. So the regulation of syndrome on those chemokines cytokines are transcriptionally on the transcriptional level. So in this part, we can delineate that in response to neural injury, someone pathway is engaged. That signal to DLK, JNKs, phosphosyndrome, and elicit upregulation of those chemokines and cytokines in the neurons. And we have data to show that this pathway actually promote the pain, the neuropathic pain. Well, as I mentioned in the beginning, there is one cytokine called IL-6, which is upregulated the neurons. But when the neurons are cultured in vitro, we cannot see upregulation of IL-6. So basically, the data is like that. So in vivo, when we introduce the injury, the DRG in the tissue, we can detect IL-6 induction with time going on from 0 to 24 hours. And we can also use ribotag to pull down RNA from the neural cell bodies, which also show the enriched IL-6 in the neural cell bodies. So basically, the neurons can upregulate IL-6. However, in vitro, when we introduce axon injury in vitro using purely cultured neurons, we cannot see upregulation of IL-6. So we hypothesize that maybe the neurons actually respond to components from other cell types. So in response to injury, other cells signal to neurons. So neurons can then initiate the IL-6 transcription. So we test this hypothesis by cultured neurons and then added the tissue lysis. So in this case, we actually homogenize the tissue lysis from the mice, then added the tissue lysis to the cultured neurons, try to reconstitute the IL-6 production. So actually that confirmed our hypothesis that really the non-neuronal cells provide us a signaling molecule to initiate the IL-6 production. So when we added the sciatic nerve lysis to the culture, we can see that there is a time and a dose-dependent manner that we can see the IL-6 is induced in the neuron in this case. So this allows us to identify the IL-6 inducing factor in vitro So we use the biochemical fractionation. So in this case, we used a few steps of fractionation, like anion exchange, ketone exchange, and the size column. In this case, we can follow the IL-6 inducing activity from each fraction. So lastly, we did a mass spec on the fraction with IL-6 inducing activity. We actually identified that molecule as CNTF. When we went back to look into each fraction which can induce IL-6, we can actually see CNTF is quite highly correlated with IL-6 inducing activity. So CNTF is indeed the molecule we pursued to which induce IL-6. And when we add CNTF wild type or CNTF mutant form into the culture neuron, we can see that there's dose-dependent response of the IL-6 inducing activity, and also there's a time-dependent as well. So we made a CNTF knockout mice. In this case, we can detect the neuron, the tissue highly express CNTF. And also on ELISA, we can see each milligram of protein, there's really more than 200 nanogram of CNTF protein in the tissue. And we can see that CNTF is actually expressed in the Schwann cell. So the expression of the CNTF in Schwann cell has been demonstrated early. So it can co-localize with S100 in this case. So CNTF is really essential for IL-6 induction. So when we add the neurolysis from CNTF knockout mice, we can see that induction of IL-6 is really gone. And when we introduce neuroinjury into the animal, we can also see in the CNTF knockout mice, IL-6 induction is really greatly compromised. So the CNTF actually signal to its receptor complex composed by leaf receptor, GP130 CNTF receptor, then initiate the STAT3 signaling. So in this case, when we add recombinant CNTF to the culture neuron, we can really indeed see the signaling events is triggered, leading to STAT3 phosphorylation. And also the expression pattern of the CNTF receptor and the CNTF in the tissue really supports the signal from CNTF to CNTF receptor. And we also use the CNTF receptor shRNA. We knock down CNTF receptor in the neurons. We can see that this response to CNTF is specific. So when CNTF receptor is knocked down, the STAT3 phosphorylation induced by recombinant CNTF is gone. So the in vivo, we can also see in response to neuroinjury, the STAT3 phosphorylation in the neural fibers is greatly compromised in the CNTF knockout mice. So STAT3 actually directly regulates the IL-6 transcription. So in the STAT3 heterozygous animal, we can see the IL-6 induction post the cytokine neuroinjury is reduced. And on the transcriptional level, the STAT3 chip-seq actually identify a few binding sites of STAT3 on the IL-6 promoter and the enhancer region post the CNTF treatment. And those regions actually contain the STAT3 binding sites, TPCC and GGAA. So basically, STAT3 directly regulates the IL-6 transcription. Well, then we start to ask when CNTF induce IL-6, where IL-6 may signal to. So IL-6 actually similar to CNTF. IL-6 also induce STAT3 phosphorylation. So we asked whether actually IL-6 can directly function as an autocrine manner to further enhance the STAT3 activation in the neurons. But that is not the case by our study. For example, when we delete IL-6 receptor in neurons, we didn't see any defect on IL-6 production. And also when we delete IL-6 receptor in neurons, we didn't see any defects in STAT3 activation in neurons. And no defects on STAT3 activation in the IL-6 knockout as well. So basically, when we don't have IL-6, the neurons can activate STAT3 normally. So then we looked into where IL-6 may signal to further far away from where the neural cell bodies are located. And in fact, if you look at the anatomy of the sensory neurons, they actually project to the central nervous system. So we looked into the IL-6 protein across the whole, where the projection route of sensory neurons. By using ELISA, we can see the IL-6 protein can be detected on peripheral nerve here, on this region, DRG, dorsal root, and also spinal dorsal fold. So basically, there's a possibility that the IL-6 protein can signal to the central nervous system. And this upregulation of IL-6 is dependent on CNTF. So in the CNTF knockout mice, the IL-6 protein upregulation across the whole rows is actually greatly decreased. And when we look at the IL-6 receptor in the central nervous system, actually the expression pattern of microglia supports the signal to IL-6. For example, the IL-6 receptor can be detected expressing in the microglia based on the single cell RNA data, those published the single cell RNA data. And we also can see the IL-6 receptor expression in microglia based on the RNA profiling data generated by Dr. Barberi's group earlier. So then we look, so the STAT3 phosphorylation in the central nervous system by the peripheral injury has been observed early by many groups actually. But in this case, we think this is initiated by CNTF. So when we look at the STAT3 in the central nervous system, we actually can see the STAT3 phosphorylation can be detected in microglia. So microglia is labeled by CX3CR1 in this case. So we can really detect phospho-STAT3 activation in the injured spine, in the injured tissue. So when we delete IL-6 receptor in microglia driven by CX3CR1 CRIG2, we can actually see that post-injury, the STAT3 activation in the microglia is actually defective. We can also use quantification to show that microglia STAT3 activation really depends on IL-6 signal. So we can verify the whole signal process from CNTF to IL-6. They activate the STAT3 activation in microglia. So for example, in the CNTF knockout or STAT3 heterozygous animal or IL-6 knockout mice, on the spinal dorsal hole in the spinal cord, the STAT3 activation in microglia or the total STAT3 activation, they are all compromised. And this axis actually can promote neuroinflammation in the central nervous system, which is indicated by microgliosis labeled by IB1. For example, in CNTF knockout mice, the injury-induced microgliosis is actually greatly decreased. And also in the STAT3 heterozygous animal, the microgliosis is also decreased as well. And IL-6 knockout mice or IL-6 receptor knockout in microglia. So basically, the injury can induce the microgliosis, while when the whole signal axis from CNTF to IL-6 is deleted, the microgliosis is reduced. So also lastly, we adopted the chronic constriction injury model because this model can allow us to assay the neuropathic pain. So in this model, we introduce a chronic constriction injury to the peripheral neuron, then we detect the pain in the central. And then we can also see this model actually can also induce IL-6 by analyzing the IL-6 production in the neural cell bodies. And in this way, we can assay that whether the signal from CNTF to IL-6 actually contribute to the nociceptive sensation by using a mechanical pain or a thermal pain. So this is the pole withdrawal threshold measurement. So in this regard, whenever you see a curve shifting up, that means the pain is relieved. So if you look at the CNTF knockout or STAT3 heterozygous mutant, you can all see the curve shifted out when this signal axis is deleted. That means the signal axis actually promotes the pain sensation. That also the same is true for IL-6 knockouts or microglia IL-6 receptor knockouts. So we all detect elevated pain when we get rid of the signal axis. So also we can detect, and lastly, actually the CNTF recombinant protein is sufficient to initiate the whole signal process. So when we apply a recombinant CNTF onto the peripheral nerve, we actually can see STAT3 activation in the peripheral neuron. So in this case, we can see the recombinant CNTF actually increase the STAT3 activation in the peripheral nerves. And in the central, the recombinant protein, when we apply to the peripheral, we can also see the STAT3 activation in the central nervous system in the microglia. So basically, exogenous CNTF can initiate the whole signal pathway, then transduce the signal to the central to enhance the pain. So lastly, the pain. So the CNTF recombinant protein can enhance the pain. So this way we apply a recombinant CNTF protein to the animal, then measure the pain in the following through few days. there's a really transient enhancement of the pen. You can see that the curve move down when we apply the recombinant CNTF. And this is also dependent on the IL-6 signaling pathway. So we use the few knockout mice, IL-6 knockout, as well as others to show that really CNTF recombinant protein can initiate the signal process and then promote the pen sensation. So this is the summary for the last part. So basically, in response to peripheral nerve injury, the Schwann cell released the CNTF, which signaled to the sensory neurons. The CNTF initiates the signal pathway to activate the STAT3 phosphorylation. STAT3 then transcriptionally upregulate IL-6. So IL-6 protein can signal to the central nervous system, which promotes the STAT3 phosphorylation. So this is the injured side STAT3 activation in the spinal cord in compared to the uninjured side. So that response is quite local. So only on the injury site, you will detect this response. So we think this could be the way the peripheral damage can be transduced to the central nervous system to alert the central nervous system there is a tissue damage or danger in that region. So lastly, I'll summarize what I have showed on the second part. So we think there's an intrinsic neuronal innate immune response to nerve damage. And also there's a extrinsic neural damage mediated by Schwann cell derived CNTF. And we also try to propose CNTF as a neural danger molecule, which is very specific to the neuron system. So the Schwann cell use this danger molecule to alert the whole animal that there is probably danger around. So lastly, I'll thank the institutional support from the Institute for Immunology, for Medicines, for the Center for Life Sciences. And I would also like to thank our collaborator from Shanghai Jiaotong University, Renji Hospital, Guang Lei, Xia, and Wenwen Zeng. And also, particularly, I would like to thank the students who have been working on the adipose tissue, Hao Chen, Xiaofan, Xia, Ximing, they have been working on the adipose tissue, and Hao Chen is now in Tsinghua University. And also, I would like to thank Zhongsheng, Nan, and Shan for working on the sensory neurons, and Zhongsheng is in Nanjing Chuanqi company now. And also, I would like to thank the funding source to support our research. So that's it. Thank you for attending the seminar, and I'm happy to take questions. Thank you.
Thank you, Wenwen. That was great and a lot of information. Okay, so I think we can start with the question. Shelley, I think you go first.
Okay, hey, Wenwen, this is truly fantastic. Like so many interesting results, and everything is crystal clear. So I fully enjoy it. So I think I can start with the last part. So this is a question actually I have never thought about, like the signal transduction and cytokine chemokine release in neurons in this highly polarized cells. So because like if for normal cells, like the signal transduction can be just mediated by like diffusive like mechanism. So I wonder, like, for example, if you can go back to the summary slides you have for the second part. Yes, this one. So for example, the STAT3 signal, so from the injury site to the cell body, do you think there's any active like a transport mechanism that is involved? I guess a related question is when these cytokines or chemokines are released, are they released like randomly, or are there any specific site that they are released? Because you definitely show like there is effect in the central like nervous part.
That's a great question. So I don't think I have full answer for that. So for first question, like the transportation of the molecules, actually in response to the peripheral neuron injury, those transfer factors are activated locally. So there is a retrograde transportation mechanism. Actually, DLK has multiple roles in neurons. So one way it leads to JNK and the syndrome activation. The other activity mediated by DLK is actually mediating the retrograde transportation of other transcription factors to the neuron cell bodies. So with time going on, you actually can see the STAT3 phosphorylation of phosphorylated STAT3 on the peripheral region, then proximal to the neuron cell bodies, then within the cell bodies. That can be blocked actually by blocking the retrograde transportation. That is the active transportation process. But regarding the release of the cytokines, we actually don't know for the time being. So we try to understand using like culture neurons to see whether we have any ways to, for example, viralize different regions of the neural cell bodies to see, for example, whether there's any coupling with the neurotransmitters or as well as other molecules as well. I don't have an answer for that unfortunately, yeah, but I would love to know actually. Yeah.
So for example, has ever anyone see like these, let's say the transport of these cytokines or chemokines along the fibers, let's say?
So it is known it can transport it by using like conditional knockout. But I don't think people have seen like using real time, for example, imaging.
I see.
So you can really see it is being transported.
Okay, cool.
Yeah.
Yeah. Okay, super nice. So the other question I have is for, I think you showed the nerve, the first part, the sympathetic neurons cover the adipose tissues. So like in normal conditions, like 90% of the adipose sites are covered by the neurons. So then my question will be, if there's already 90% is covered, why do the neural network still extend like during the cold treatment? Does that suggest it's trying to reach out like other type of cells, like for example, like immune cells, in addition to the adipocytes?
That's an interesting idea. We didn't see, for example, so sympathetic neurons are not like motor neuron or other cell types. They don't really form those synapse structures to signal on a one-to-one manner. So it's more like a one-to-multiple manner. So one possibility is the strength or amplitude is increased. One possibility is really can reach out to many more cells in the tissue. Yeah, but that's the interesting idea. Yeah.
Okay, cool. Maybe last question is, I'm sure you're also thinking about like the cold-induced regeneration and angiogenesis is largely compromised in like obese and the diabetic like conditions. What do you think is the mechanism? Like, what's the difference between the normal and the pathological conditions?
Yeah, that's a good question as well. So under pathological conditions, there's actually, I think it's not a single mechanism because the environment is really not friendly to the survival of the nerve fibers. So one possibility we test is whether NGF level is changed. Actually, it's not. So there's really pathological molecules which actually not single molecule, which many factors actually can contribute to axonal death in this regard.
I see.
So there's actually multiple, for example, inflammatory cytokines can actually contribute to that. And also as well as a survival, the survival is not very well supported as well. Yeah.
Did you see any like cell number change of, let's say, the CD45 positive cells when treated?
Cell what? I'm sorry, I didn't hear.
The CD45 positive cells, did you see any change in the CD45 positive cells during the cold treatment?
Oh yeah, we actually working on that for the time. So in response to cold challenge, there's really immune microenvironmental change. So it's more like type 1 to type 2 immune response change. So there's so the type 2 immune signal axis is actually involved in that process. Yeah.
Oh, cool. That's very nice. Yeah, fully looking forward to your future like exciting results. And thanks so much.
Thanks, Shirley. Thanks a lot.
Coco, you're up.
Hi, Wenwen 老师.
Hi, Coco.
Very nice talk. So my first question is sort of follow up Xiaolei's question, his first question, I think. So microglia is known to be one of the sources of IL-6, right? So could it be like in your experimental settings, the like microglia derived IL-6 also play a role, or also like the expression level also change it after the tissue damage, and there might be some sort of autocrine?
That's interesting. But I think so. We used so the very unfortunate thing is we use the IL-6 global knockout. So I think that might be able to address by using conditional knockout mice, because it's really hard. So maybe if you use like in situ other ways, we can determine whether the IL-6 mRNA is really distributed to the axons or microglia, because it's possible that microglia also upregulate IL-6 in this process, forming like autocrine loop as well. I don't have an answer, but it's highly possible actually.
Yeah, I know that the IL-6 phosphorus is not commercially available, right? So it might be a hard experiment to do. So my second question is also about the second part of the talk. So have you ever tried a spinal cord like contusion model, like similar to the traumatic brain injury model, but applied onto the spinal cord? So now the model you use is like the fiber from skin to DRG. I was just wondering whether you have a look at like the fiber from DRG to spinal cord.
Yeah, we didn't do that. So the distribution of the Schwann cells are quite specific. We didn't do that part.
I see.
It's possible that they are different, because the distribution of Schwann cells, so that because in the peripheral, in the sensory neuron, is the myelination of the axons are quite different on the dorsal root and the peripheral nerves. Right. But that actually is an interesting experiment to verify the role of the dorsal root.
So just one last question regarding your first part of the talk. So we know that stress can induce norepinephrine. I was just wondering whether stress could like directly, I mean, through norepinephrine release to influence Beijing and lipolysis.
That's an interesting point too. So it's possible that, so we didn't use stress model because stress actually can induce other like stress molecules which have opposite effects of
it changes a lot of things.
So we didn't use that model. So we think the code-induced Beijing is more of the engagement of local sympathetic neurons, but stress can more like engage the adrenal gland and releasing many of the molecules from there. If we can dissect these two like branches, we might be able to separate the effects from the sympathetic neurons as well as the hormones from circulation. But under some of the conditions, we had some collaboration with other groups. It's possible that you might be able to engage the sympathetic neurons under stress condition, which can drive the Beijing under the pathological conditions as well.
That's super cool.
Or
maybe like also like optogenetic like experiments like turning on some stress related brain regions and I don't know, just sort of like brainstorming and the like I think is a quite interesting question whether like people under stress whether they gain more brown fat or I don't know.
Or if you want to lose fat, you engage some of the brain nuclei, so you can actually burn fat as when you want. I think it's possible you can actually, because when we look at the projection of the fat from the central nervous system, you can actually see the brain regions in the hypothalamus, which has been shown by a few other groups as well. So if you manipulate the brain nuclei in the hypothalamus, it's just, I'm not sure how like translational that is, but you can certainly drive the process from the central nervous system. Yeah.
Cool. Thanks, Wenwen 老师.
Thank you, Coco. Nice to see you.
Nice to see you too.
Okay, so I think Yuchun Wang has a question. You want to ask you about self? All right, so I'm gonna just read the question. So I think first question is whether the parasympathetic neurons also innervated into the adipocyte.
Okay, yeah, a parasympathetic.
Yeah, parasympathetic.
So in mice, we didn't see many parasympathetic neurons in the adipose tissue. So in the, I think that part has controversy in different groups. So one reason is different model animals, for example, hamster, rat, and mice have been used in different groups. It is possible some animals may have different anatomy, but in mice we didn't see many parasympathetic neurons innervating adipocytes.
What kind of marker are you going to use if you do want to check the parasympathetic?
We actually use the chat Cree reporter mice, and also WeChat immunostaining.
Okay.
Yeah.
I think he asked the following question is, you have the cold-induced thermogenesis which relate to the sympathetic neurons. He asked the consequence of the adipocytes and the innervated nerves if the mice are exposed to hot environment. I don't know if you can do that.
Oh, yeah. Well, the highest temperature we went was 32 degrees, the thermoneutral. So.
Okay. Did you do that?
32?
No, yeah.
32 is fine, like thermoneutrality. Yeah. Well, the innervation is decreased. Yeah. In response to warm.
I see. Okay.
Yeah.
I think the second part he asked that what is this function of the recruited immune cells in the axon injury?
Well, that's a good question. Actually, I don't have an answer, but we have some speculations. So we think that might mediate the immune remodeling to promote the regeneration of the axons. So we have a technical challenge in this part because we had difficulty to deplete the cells locally. So whenever we deplete the cells, we manipulate the cells, we manipulate the cells on a systemic level. Because on systemic level, actually the CD9B positive cells are crucial for axon regeneration on the axon injury site. So we still try to separate this part, because from the anatomical location, what are the functions of different subcells in different anatomical locations? Yeah, that's that.
Okay.
Yeah.
I think the following question, Jean actually has a question that I think that's overlap with the previous one. He asked that what happened if the body temperature increase instead of, but I guess he's probably more mentioned like in the condition like fever. But you're saying that the innervated nerves gonna have opposite effect as the mice under the cold condition, right?
Oh, like a fever. That's an interesting question. I actually don't know.
Can I ask myself? I'm the Jing.
Yeah, go ahead.
Okay.
Well, actually, it's not fever. Like, if the mice they're hyperactivity, so I don't know is this also cause higher body temperature? Because I noticed about a lot of mice, if they have hyperactivity, they're pretty skinny. So I don't know, because I'm working on the neuroscience. I don't know, is there any relationship? Because you said in a cold temperature, there increased the neuron and angiogenesis. So what would happen, like the sympathetic neuron, if they have a hyperactivity? So what will happen for the fat? If the fat reduce the fat, or?
You mean like the core temperature change? I wouldn't see the core temperature would change. You mean the hyperactive mice? I mean, they, for some reason, for many different reasons, they're hyperactive.
Yeah, they have some hyperactivity. So you said, but you mentioned about if increase temperature, they will cause loss of nerve and angiogenesis. That's true.
Yeah.
Okay. So have you ever, actually it's another question. Have you ever monitored the sympathetic neurons activity? Like if they are hyperactivity, what will happen to the, you're starting the white adipose tissue? What will happen to the fat? It will cause induced fat, reduce the fat, or so it's a neuron. Yeah.
You
Like
mean if we artificially active the sympathetic neural activity?
Yes.
Or that can induce the Beijing. So we have used actually optogenetics and chemogenetics. We have done that. That can induce Beijing. Yeah.
So what is Beijing? I'm not in the field, so can you explain to me?
So the white adipose tissue will become darker because the cells are generating more mitochondria. So they become darker, so they can produce heat because they highly express they highly express UCP1. It decouple the ATP generation from the ETC in mitochondria. So that actually leading to heat generation.
So the heat generation, it means you're burning your fat.
Yes. So in response to cold, actually it used the lipid as the energy source, one energy source. Yeah, okay. To generate heat. Yes.
So that's if increase the sympathetic neurons activity, you can start the Beijing and also cause they transfer to the brown fat and they're just kind of lose their fat. Can I see that? Can I get a conclusion?
Yeah, quite close. Yeah, so you have the white adipose tissue Beijing and you have the lipid consumption, so to maintain body temperature. Yes.
Okay, that's very cool. Thank you.
Thanks.
Paul, you want to go ahead?
Hi, yeah, for sure. 谢谢传，谢谢郑老师。It's a very nice talk. I really enjoy it. I just have a general question about the microenvironment of nerve and then monocyte or macrophages. Because based on your data, you showed that DOK or June knockout, the CSF1 is reduced. And then I believe that will affect the monocyte macrophage. And based on what I learned, paper probably last year, people showed that NatureCom paper, they showed that the peripheral macrophages show a similar gene signature compared to the microglia. And then Martin Gilliams later on showed that under the injury conditions, I believe this injury condition probably is similar to what you were doing before. And then they showed that the monocyte macrophage actually came in, but without showing any function. So my question is that in general, can you comment about what happened in your condition in this DLK knockout or June knockout during injury? What happened of macrophages in that microenvironment?
Yeah, that's a good question actually. So there are like list of cytokines and chemokines which are regulated by this pathway. So some of them actually have been shown by, for example, Dr. Ji's group. They show that some of the, for example, some of the like and as well as other groups as well. So CCL2, CKO in the sensor neurons, or CSF1, CKO in the sensor neurons, that actually can influence the microgliasis in response to injury. So that is how the sensor neurons actually they upregulate those chemokines, cytokines, influence the central nervous system. If that's what you mean. So I think CSF1 can have multiple roles. One way it can influence microglia. The other way it can certainly influence the macrophage in the peripheral the microenvironment as well, because it can influence a few cytokines, CCL2, CSF1. So it can, in the combination, it can either influence the amplification, the proliferation or survival of those macrophage, and also as well as microglia. In DRG, there are also the cells could be influenced as well, actually. And also in the injury site, because in the injury site, you would also have like macrophage and monocyte and macrophage differentiation to support angiogenesis, because you need the axon to regrow to the terminal region. So CNTF can also play a role in that part too. But we didn't do that like experimentally. I think that is based on like the previous evidence and the role of CSF1 that can certainly happen in all those places, I believe. If that's
Yeah, thanks so much. Yeah, I'm just curious because based on the previous observation, 2 papers show the peripheral macrophages behave like microglia, but after injury, the gene signature is gone. So I'm just try to relay what they found to your studies, and hopefully to understand the upstream and downstream relationship. So yeah, you did give me some clue and answers. Yeah, I'm looking forward to know more about what's going to happen in this field. Thanks so much.
Thank you.
Okay, so my turn. So
all right, you have questions?
Yeah, I have questions. Yes, of course. Yeah. Well, I have first a comment. So actually, I think I want to just address what Xiaolei asked you first. So I guess his point is when you produce the cytokine chemokine, where it's actually expressed, right? So why it's from the body but not from the exosome? So I think there's literature report that in the neurons, because they have really long exosomes, so their lysosomes will actually bind to the RNA granule and then protein granule. So we'll transport, the lysosome will go along the microtubule in the neurons and then transport these materials to the target location. For example, if your IL-6 be generated from the body, they usually have lysosomes bind to these IL-6 and then transport it into the central nervous system along the microtubule in the neurons. So that's how they actually go to different locations. But usually these are related to very small proteins. I'm not sure how big the protein of the IL-6 will do the similar behavior. It's a unique cellular behavior only in the neurons, but not on the other, because neuron has this particular morphology.
So thanks, Trev, for mentioning that. I think that's like probably referring to like that paper by Jennifer Pinkow-Schwartz. So
yeah, I mean, there's a couple of different papers particular describe that protein and then RNA granule being transported within the neurons.
Yeah, so these are hijacked, especially for these things, the recent Cell paper. But most of these cytokines and chemokines are pretty small molecules, which they don't have these extensive, let's say, intrinsically disordered region or these like region that adaptive protein has. So that become an interesting question, I think, like how do these like very simplified like molecules can hijack the transportation system.
That's true. I mean,
and also I think my other point here is like, why doesn't it just release? I think when we're trying to address this, like, why doesn't it just release on the cell body? Or is there any specific like location it designated to so that it's specifically released at that location? So I think that one is, so if that's the case, then we need to think about the transport system. And it seems that's true, at least for the IL-6 case. So yeah, I just think there's a lot of interesting like cell biology questions here, which I haven't thought about before, and thanks to Wenwen's talk today. Okay, sorry for the.
No.
That's really good to know. So, but for those chemokines and cytokines and many small molecules, I wonder they actually use a hijack system instead of like a regulated process or just a passive diffusion, because that sounds really not very efficient.
Exactly, because the passive diffusion, some people have made a calculation, it takes forever to reach the, and also in most cases, these cytokine, chemokine not expressed in such a very high amount. And So, or maybe I will just say like they are like encapsulated in some like specific vesicles.
Yeah, that's maybe yeah. I kind of speculate there maybe some sort of like a granule or vesicle-like structures they actually carry on for the transportation.
Yeah, so then they have like a regulated release at specific site.
Okay.
Okay.
I think I think the target, like if the IL-6 will go to the spinal cord or the central nervous system, like you say, they must have a signal circuit which actually guide these specific molecules from the body being synthesized and then transported to there. I'm just to state that there's potential machinery which actually can be direct by the microtubule on the lysosome,
that's possible.
and then to transport them to there. The question is why? I mean, why there's circuit which actually from the neuron body through the exosome into the central nervous system? I think that's an interesting question. You probably will be able to look into. I think I have a lot of questions. I have a list of questions, but I only have two want to ask you. One is, you say that the TRKA actually during the thermogenesis, you see the UCP1 get transcriptionally upregulated. My first question is why? Why? So why the signal, the code signal through the TRKA will upregulate transcription level of UCP1? What's the potential signaling pathway? Or it's maybe been previously published. I mean, because you see the both protein and the mRNA is increased, right?
You mean the adipose size?
Yeah, in the adipose size. Yeah.
Track A is in neurons.
No, the TRAP-A knockout, you see that under cold condition, the UCP1 is not in the adipocytes, not changed, if I remember correctly. Maybe I remember wrong. You talk about so many things. So
I think you probably messed two slides together. So UCP1 is upregulated, that's for sure.
Yeah, so I think I might miss some link there. So my question is, the thermogenesis or the cold signal through the sympathetic neuron to induce UCP1 transcription is through what? What's the substrate?
Oh, all right. So that has been studied by a very long time, actually.
So what is the substrate?
The transcriptional level.
Yeah.
So there's some transcriptional mechanism which have been proposed, because if you think about the signaling events actually from the adrenergic receptor to UCP1 upregulation. So from adrenergic receptor, people speculate is CRAB. So CRAB can certainly but CRAB is not sufficient. So there's also other transcriptional factor involved, like PDC1 alpha could be one transcriptional involved cofactor.
Okay.
Alpha gamma.
Okay. So the other thing is when you talk about second part, that you have the SARM1, DLK, JUNK, and the C-Jun signaling pathway. So any of these knockout actually can cause quite big neuron deaths. Have you seen that? I mean, for example, the JUNK knockout, you see the reduction of the neuron body.
Right.
Have you see that? I mean, because all the phenotype you see from the neural injury that reduce all these chemokine, cytokine, and they're probably related to all these signaling pathway, because most of them are actually kinase. If you knock any of these elements out, it will cause neuron apoptosis or neuron death, and then that's probably also induce reduction of the what's so-called immune response. So
yeah, you're right. So there are mixed. So I think one clear gene is SARM. So that so developmentally, it doesn't have effect for like neural apoptosis. But for junk, so junk has like junk 1, 2, 3. So junk 1 is actually quite essential for the survival. So we wouldn't for the animal to survive. So but junk 2 and junk 3 double knockout, we didn't see defect. Like the neurons, I mean, under homeostatic condition, they're fine. But for Cedron, it's true, they cannot survive. So that's the reason we only have in vitro data. So we transiently introduce Cray, and then we introduce injury. Because that can cause both. That's true, because a lot of genes actually have roles on cell survival. So that is a challenge actually when we make the claim. It's kind of hard to based on one gene, because some of the genes are really essential. Yeah, like Cedron is really essential for survival. So we wouldn't like based on Cedron to say that the pathway is really essential for the neuroimmune response.
Because then it's actually leave a quite obvious caveat that your signaling pathway, you proposed signaling pathway, it's hard to demonstrate that this is a linear signaling pathway, because some of them are so important that you won't be able to demonstrate these are the the main or the essential one, but not the sufficient one. So
it is a challenge for anything downstream of SARM actually. For SARM is quite unique, but downstream of SARM, everything is shared between many of the pathways, especially the stress and the survival. Many pathways actually converge on the downstream pathway.
Have you ever thought about, because I feel it's actually quite surprised that it has to go such a convoluted signaling pathway, goes through so many different elements, nodes in the between. Why the SARM1 cannot somehow more efficiently direct control all these downstream, signal? I mean, maybe 1 or 2, maybe, but what you propose is SARM1 through DLK and junk and CJON, and then to the target. It's actually very, very long. Many stop in the between. So I was just curious, is that the real case, or is there somehow shortcut which actually can more efficiently control the immune response?
Yeah, it's so even for someone, it's actually someone mediates in a real situation, someone actually mediates the energy consumption in the axon cell body to mediate the axon degeneration. So I think this part, the reason it's very complicated question. I don't think I have a good answer for that. But I think there are many effects someone want to achieve. So one way is it want to alert the central nervous system of the damage caused by peripheral that is induced by the neuroimmune response. But the neurons have to decide whether it wants to survive or die, because that's the way it wants to protect. Because axon degeneration is actually a protection mechanism sometimes.
Sure.
So it's not necessarily true that survival is an advantage for some of the neurons in response to damage.
That's
so
true.
Maybe it has to carry 2 pathways in order to determine if I die, I have to tell the central nervous system I want to die. But that is carried by the same molecule downstream.
Yeah, but that's actually something you can think about, because obviously neuron also carry the other common innate sensors like mediated or TRIF.
Right.
Not only the SAM1, although the SAM1 is the unique one for the neuron, but there's must be somehow a reason behind it why it's actually goes through specific to SAM1, but not actually go through mediated one, 88 or the TRIF one.
Yeah. So someone seems more like deal with the structural change and energy state.
In the neuron.
Yeah, mediated and TRIF more like deal with the like foreign or internal infectious agents.
I see.
That's more like diverted on the roles to detect to deal with the threats. I guess. Yeah.
Oh, that's interesting. Okay, well, I'll let it go so I can ask you afterwards.
I'll leave the 1 page for next time.
Okay, so if no one else has questions, thanks again to Wenwen's great talk. I think it's great discussion. And then hopefully I see you guys next week to the immune seminar. So thank you very much, Wenwen.
Thank you, Chuan. Thank you, everybody.